Hello, everybody. Welcome back to our discussions about blood flow and pressure. In this video, we are going to be talking about capillary exchange. All right, so I want to just start off by telling you a little bit about capillaries, or a little bit more than you've already heard. As you know, capillaries are the site of materials exchange, right? So this is, in my mind, this is kind of where all the action is, right? The whole point of the cardiovascular system is to get things to different places in the body. And the capillaries are where the exchange actually happens, right? So the arterioles, the arteries, the veins, all those other blood vessels, their job is to get the blood to and away from the capillaries so that that exchange can happen, okay? A little bit about capillaries. First of all, we have lots of them and they are very densely distributed in our bodies. So all the tissues in our bodies are within 0.1 millimeter of a capillary. And that kind of speaks to their function, right? So again, we are delivering materials to different areas of the body. We want those areas of the body to have access to those materials, hence their proximity to a capillary, right? So any tissue can access the capillaries quite easily, okay? I do want to mention that the density of capillaries varies in different parts of the body, depending on those the parts of the body's needs for the various nutrients, right? So remember, a good example of this is, remember when we were talking about fast twitch versus slow twitch fibers in the muscle, the slow twitch fibers needed a higher density of capillaries to bring them oxygen to do their oxidative phosphorylation, whereas the fast twitch fibers didn't need as high of a density of capillaries because they depend more on glycolysis for their metabolism. Finally, I want to say that the capillaries have thin walls to facilitate exchange of materials, right? So remember that we just have a single cell layer thick layer of endothelium that makes up the entire capillary wall. And since it's just one cell layer thick, that provides a nice surface across which we can exchange materials, right? So we can have diffusion happen across there, or transcytosis. We're going to be talking about those in just a little bit. Okay, so that thin wall makes it easy to move materials from the blood into the surrounding tissue, or vice versa. Okay, one more thing I want to say about capillaries is that we have a low velocity of blood flow in the capillaries. So I didn't really go into like velocity of blood flow and its relationship to area. So don't worry too much about what it says right here. But basically, the blood is flowing pretty slowly through the capillaries. And that is great because that then allows plenty of time for materials to be exchanged, again, between the blood and the surrounding tissue, or from the tissue into the blood, okay? All right, so now let's talk about how we get materials across that capillary wall, right? So we need to move materials, again, from the blood to the surrounding tissue, or vice versa, okay? We have three main ways in which materials move across the capillary wall. The first of those is diffusion. So I'm going to start by talking about diffusion, okay? Diffusion happens across the capillary wall. It can be transcellular or paracellular. So just to remind you what those terms mean, transcellular means that the molecule actually goes through the cells that make up the capillary wall, whereas paracellular means it goes in the spaces between those. So looking at this figure right here, we have a cross section through the capillary wall right here, right? So if you took a capillary and just cut it like this, you would get this, right? So what you're looking at here is, this is the inside of the capillary, this is the outside of the capillary. We can cross, right? So we can diffuse transcellularly. The way we would do that is if we pretend my laser pointer is a solute diffusing across the capillary wall, it would just diffuse through this endothelial cell, through one side of it, and then through the other side of it, and out to the surrounding tissue, or vice versa. If it were going paracellularly, it would avoid going through the cells. It would go in this space right here that lies between those endothelial cells, okay? Another thing I want to say about this is that the endothelial cell junctions, right? Remember that we have tight junctions there. They're going to vary in their leakiness, okay? So we can have some tight junctions here that really don't allow anything through. So remember that we talked about that when we talked about the blood-brain barrier, that we have these tight junctions that are very hard, that make it very hard for solutes to pass in this paracellular space. But in other cases, we have pretty leaky junctions where those tight junctions are not very tight and we can allow solutes to pass through. So that's going to vary from tissue to tissue within the body, okay? One more thing I want to say is, in addition to having these spaces between endothelial cells, sometimes we even have this situation right here where we have these kind of big pores between the endothelial cells that make up the capillary wall. And in those cases, those capillary walls are very leaky, and pretty much anything, I don't know about anything, but a lot of things can pass through those walls. Okay, so we tend to have this type of capillary. They're called fenestrated capillaries in areas of our bodies where we do a lot of absorption, such as the digestive tract. Okay, and then finally, I just want to close this slide by saying that most of the solutes that are crossing the capillary wall are crossing using this technique. They're crossing by diffusion, okay? All right, our second method that we have for solutes to pass across the capillary wall is transcytosis, okay? So what you're looking at here is this is the endothelial cell. This is gonna be the inside of the blood vessel, and then this is the outside of the blood vessel, the capillary, okay? So if a molecule is not small enough to just diffuse either across the cell or in that space between the cells, it can be transcytosed across the endothelial cell. The way transcytosis works is basically, here's our molecule that we want to move across the cell, across the endothelial wall, okay? It gets endocytosed at one end and then transported as a vesicle across that endothelial cell, and then it gets exocytosed at the other end. Again, this is for larger molecules such as proteins, and this is something that can happen either in those regular, like what we call continuous capillaries, or in those capillaries with all the holes in them, the fenestrated capillaries.